Hi, my name is Haider Abbas. I'm working as Diversification Specialist with Manitoba Agriculture and Resource Development at Canada Manitoba Crop Diversification Center in Carberry. This is a beautiful morning of August 2020, and right now I'm standing in front of a mustard study, and this mustard is seeded in standing stubbles of winter wheat and fall rye. This experiment is set up with the support of Manitoba Horticulture and Productivity Enhancement Center, and the aim of this project is to test the maximum biomass production ability of different mustard varieties for the biofumigation process. And the biofumigation process is used to control soil-borne diseases in potatoes and other crops. I have pleasure to have Zach Frederick from AMPAC with me, who will highlight the field component of this study.
Thanks, Haider. My name is Zach, and I'm your friendly neighborhood research agronomist who works for MPEC Inc. That's the Manitoba Horticulture Productivity Enhancement Center Inc. And I will explain a little bit on how the field portion of the mustard biofumigation project works.
Hello everyone. This project that we'll be talking about is the Mustard Biofumigation Project, which was initially developed in 2018 as a tool to help growers decrease the number of Verticillium wilt propagules, called Microsclerosis, in their soil to manage disease. It's important to note that we arrived at this study because for the overall field variability study, the number of propagules of verticillium found in soil was found to be one of our most significant issues for especially the larger tuber size profiles, where the more propagules you have, the fewer larger tubers you generally have in that same place for potatoes. So going back to biofumigation, it is one of the treatment options for verticillium wilt. There are other options such as fumigation, crop rotation, water management, and the specific cultivars of potato used. There is a longer list of potential options, and depending on the severity of your situation, you may need one or more options to provide verticillium management in the way that you deem most desirable. The Microsclerosis that I spoke of is a specialized term that refers to the means by which the fungus survives between potato rotations. They're a dense aggregate of the thread-like body of the fungus that are known to have a lifespan of 5 to 15 years, depending on which scientist you're getting this information from. It's important to note that for the 15-year upper span of their life, that was because the scientists stopped studying them, not because they were found to completely die out. At any rate, these structures are going to be, based on how they're constructed, they're resilient to heat, sunlight, drought, flooding, chemical pesticide, and they have a very, very long lifespan that can be both with hosts, which they have many things they can infect, or they can live that long without a host, say in a jar on a shelf in a laboratory for many, many years, or just sitting in empty soil in the ground for years. It's important to note, as I alluded to before, that this fungus has a very wide host range, meaning that your rotation crops and your weeds are also involved in the spread of disease, and even plants that don't get verticillium wilt. So research has indicated grasses in particular, we're talking wheat, corn, rye, are all hosts of verticillium, but these plants do not get diseased upon infection, but they do allow the fungus to persist, as well as the fact that it can just live in your soil throughout a whole crop rotation. Weeds like nightshades are going to be important because they really are quite susceptible to infection and they show no symptoms of disease whatsoever. So they not only allow the fungus to persist, but multiply, and possibly with nightshades, we're talking a situation where the verticillium becomes better trained at killing your potatoes by becoming aggressive over time. Going back to biofumigation, this process refers to raising a mustard crop in order to turn it under when the mustard is flowering, which is typically after about 2 months of growth. Compounds within the mustard plant react with oxygen and soil moisture to release a gas that is toxic to soil microbes. In sufficient doses over a sufficient period of time, it has been determined that Verticillium microsclerosis can be killed in experiments in Canada, the United States, and Europe. So this process has been vetted in a variety of different situations. However, research has indicated that not all mustards were created equal with regard to their biofumigation potential. Oriental mustard can be used for biofumigation purposes, but yellow and white mustards have not been found to be as useful. Also, the method of biofumigation is very, very critical to success. Biofumigation works best when the mustard is utterly destroyed by a flail rather than by disking it under or plowing it under. And also, because we're talking about the release of a gaseous compound, it needs to be buried ideally within 5 minutes, but certainly no more than 10 to 15 minutes after being damaged, or else you've lost all the gas. That will limit the success of your biofumigation. And as such, the main point of this research is to provide a list of recommendations for mustard biofumigation and for Manitobans that has been experimentally vetted. And our potato research website at mbpotatoresearch.ca has the newest version of these recommendations for your best shot at success. And every year that we've been conducting this research, we've been fine-tuning those recommendations. So it's important to check back periodically on those. And if you have further questions or comments, please feel free to reach out to me at mhpec@outlook.com. That's mpec@outlook.com. For the research reports for this present year, in 2019 we had a problem where there was significant snowfall at harvest that really prevented proper biofumigation in most fields. Also, given the wetness and the prioritization of potatoes, obviously as the money-making crop for these farms, mustard just kind of fell by the wayside. Superficially, with the 2 fields that survived last year, if you had sufficient biomass, it appears that biofumigation did reduce Verticillium Microsclerosis in the field, but really we need more years to really verify this statistically. In 2020, we have 6 mustard fields that we're following very closely, and I'm very pleased to report that with a June planting date, we have achieved the desired biomass of 5 feet of mustard plant, most of that being leaf matter. So we're really optimistic this year that we will have our best shot of success, and I look forward to sharing these results with all of you in due time. Thank you all. Over to you, Haider, to talk about your experiences with CMCDC-based research in biofumigation.
Biofumigation is the suppression of soil-borne pests and diseases through the use of plants that produce inhibitory chemicals. In most cases, these biofumigant plants are chopped and incorporated into the soil so they can release their inhibitory chemicals. CMCDC is testing different mustard varieties to develop agronomic recommendations for potato producers to generate maximum mustard biomass, as biomass and glucosinolates are factors that are fundamental to the success of biofumigation. Mustard come in many varieties, but not all are equally as effective when it comes to biofumigation. Some mustard varieties produce more glucosinolates compared to others. For example, Chianti contains very high levels of glucosinolates. At CMCDC, we are testing 3 mustard varieties: Chianti, AC Volcan, and Andante. Mustard is seeded in standing stubbles of fall rye in late spring, as we have seen more success in seeding mustard in standing stubbles. Some other factors affecting biofumigation process are seeding date, soil pH, fertilizer. Nitrogen is important to the production of biomass, and sulfur is crucial for the production of glucosinolates. Nitrogen is applied depending on the field's history. The rate of sulfur should be adjusted in relation to the chosen nitrogen rate in 6:1 ratio. When incorporating the mustard crop into the soil, mustard crop should be incorporated into the soil before it has reached full bloom. Incorporation process must be done when soil has a good level of moisture. Do not incorporate mustard when the soil is dry. Mustard must be incorporated immediately after mowing. 80% of the fumigant gas will be released in the first 20 minutes after mowing. After incorporation, the field should be rolled and packed to trap the fumigant gas in the soil. Finally, Once the incorporation process is complete, leave the field undisturbed for 14 days to ensure that all the plant material can break down. Although mustard is a remarkable biofumigant, it has similar benefits that is expected from any other cover crop. Prevention of soil erosion, recycling of soil nutrients, improved soil structure, and maintaining soil organic matter. For result of the study, please visit mbpotatoresearch.ca and mbcropdiversificationcenters.ca in fall 2020. Thank you.